big algorithms and machine learning, genetics, and a lot of previous stuff. And today we will be giving a talk on how algorithms in computer science can be applied to biology. And it is PhD in physics from Indian Institute of Science, IMSc. And during his second postdoctoral, he got interested in biology and knowledge of computational languages. So very excited for the talk.
Yeah. Okay, thank you very much, Sonia, and all of you for inviting me here. So I've never actually done anything like this before, that is speak to expose students about actual research. I won't be talking about my research, but so also I think Ananya might not have heard me speak before, otherwise she might not have invited me. But from the plus side, if you ask me questions, it'll be much better. So it's a small informal group, so please ask me whenever something is not clear, whether they want me to say more about something. Okay, so you're in. So the first question is, the title have algorithms and have computer science. How many of you program computers? Do computer programming? Most of you. How many of you know what? Have not programmed computers, but you're kind of familiar with how computer programs are written. Okay. So an algorithm is so it actually originates, I think, from Arabic at some point, but it's like a procedure. It's a way to do a task, and it need not be about programming computers necessarily. For example, you might have read Euclid's algorithm finding the greatest common factor, and so it's a set of rules that you do one by one, or so a computer is a very dumb object. You have to explain to it exactly what to do. And so computer science is like writing things formally in such a way that a computer can do them. And there's a quote from a well-known computer science textbook by Abelson and Sitzman that I like, that a computer program should be written for humans to read and only incidentally for machines to execute. So essentially, algorithms are ways to write instructions for humans to be read by other humans. And implementing them on a computer might be kind of a secondary task. But that's it. Implementing them on a computer is what we need to do to solve any of those problems. So I'll be talking about some of these problems we're talking about here. The reason, so that I'll mainly be talking about sequences. And now in real life, you have a lot of sequences. In biology, this is an example of the sequence of DNA. So if you're not familiar with this, okay, it comes with a little bit, but if you go to websites like NCBI, which store bioinformatics information, and you ask for the genome of human or equal, I think you'll get some string that looks like this, except it will be millions or billions of letters long. And that string basically represents the content of the DNA of that algorithm. So this is a string, it's a sequence. The sequence is just one symbol after another. Here you have the 4 symbols. You could have other sequences. So in the English language is a sequence of letters. And in the simplest case, you could think of it as a sequence of 26 letters and a space. You could remove all other punctuation, and you could forget about uppercase and lowercase. And you can do quite a lot that way. And but you can also think of it as a sequence of words. And both ways of looking at it are useful. And people try to model how these sequences might arise. So you all sort of large language models like ChatGPT and so on. So those are very sophisticated versions of what's called a language model, and might not really end up talking about that, but because some of those same techniques are also applied in biology. And even, for example, music is a sequence. It's a set of notes played one after the other. In some cases like this, you might have a note that it's a bunch of notes played at the same time. Whereas classical music. In Indian music, it is more linear, so you would actually have multiple notes on the same time. So music also can be represented and analyzed using some of the same techniques I'm talking about. And even spoken language, speech, when you speak, you don't think of it in terms of letters, but rather in terms of phonemes, so the word persons, sounds, and a computer can kind of learn to break that up, and computers are very good at that. You can talk to your phone and select language to, I don't know how to do that for you. So the state of the art is really advancing, but we are talking about biology here. Okay, so what's common to all these sequences? Basically, all of them can be written as a linear form, and they are all drawn from a discrete finite set of symbols, which is called an alphabet. So in ordinary English language, alphabet is 26 letters. The alphabet of DNA is 4 letters, A, C, G, T. The alphabet of proteins is 20 letters. We won't talk about that. And they can be very long, and they contain some kind of a meaning. So just a random list of letters from A to Z does not give you any meaning. And actual English language is very non-random. E is very common, vowels are more common, Q is less common, that kind of thing. At small scale, they have correlations. For example, in the English language, you often have an E followed by another E, or an E followed by an A. But in English, you don't have A followed by A very often. You don't have a double A very often. Whereas in Dutch, you do. And it depends on what language you're talking about. And longer scale, they're uncoordinated, meaning if you look at this, they can be written as what word is likely to come after written as is a fairly likely word to come. That's what you mean by correlation. If you know one word, you might be able to predict the next word and maybe the next word. That's what the language models do, but you might not be able to predict 7 or 8 words down the line. So the correlation kind of go down like that. And remain more common features. So I'm just motivating why you can think of biology as a sequence similar to language, and then you can use some of the same techniques that have been developed for language to study biology problems. Okay, so DNA, I wrote it as a sequence of letters, but as you all know, it's a double helix. This is a picture taken from the book by Albert et al. And but it's a picture you all know. So essentially, what DNA consists of is a backbone that consists of a sugar, which is called deoxyribose, and a phosphate group, deoxyrine group. You've all done high school chemistry, so these things which are called bases. And there are 4 bases in DNA: A, C, G, and T, which are adenine, guanine, thymine, and cytosine. And these bases attach to the sugar phosphate group to form what's called a nucleotide. And the nucleotides polymerize. That means that they can stack on top of each other. The phosphate group can hybridize with what's called a hydroxyl group on this side, a 3' hydroxyl group. And that means that each strand of DNA actually has a direction. That this is called a 5' strand because it's got a it's got a 5' phosphate group in it. This is called the 3' direction because it's called the 3' hydroxyl group in it. And the thing about these nucleotides is that they like each other in pairs. That is, G likes to bind up with C, A likes to bind up with T. So when you have 2 single strands of DNA, they very quick, and if the pairs match, so if you have G, T, A, C on the side, but C, A, T, T, G on the side, they will instantly kind of match up and form a double-stranded thing, which in turn forms this helical structure, which is what we all know. So a couple of things that I wanted to note. We showed in the previous slide a single sequence like this. but in fact, there are two sequences. And the reason we report one, of course, is that if you know one, you know the other. But also, the two strands go in opposite directions. Like if this is the 5' end of the one strand, you conventionally read it from the 5' to the 3' end like that. So you will read this set of letters, T, A, T, T, G, whatever. But you could equally well read the other strand, but you will have to read it in the opposite direction because here the 5' end is on this side. So you'll have to read it like that. So, and both of these mean exactly the same thing. So your genome, if you take, let's say, chromosome 1 of human, it's kind of your choice whether to read one strand or the other. If you have one strand, you can reverse it and complement it. It's called reverse complement, so that from the sequence you can get back the other sequence. So maybe that's the simplest algorithm we can start with. Given a sequence like this, A, T, A, G, C, T, A, what would be the sequence of the complementary strand? And that's called the reverse complement because you have to reverse it, you have to read it in the other direction, and you have to complement it, meaning you have to replace A with T, G with C. So you will get A becomes T, A, G, C, T, A. That's how you would read the other strand. Of course, the way it is paired here, the T will be here, the A will be here, and so on. But if you read this strand this way, you will have to read the other strand this way. So that's one thing to know because so DNA has all kinds of significant information in it, like normal language does. The one thing that everyone knows about is genes. So DNA has stretches of sequence that are so-called genes that code for information, mainly they code for proteins that you can make. And that information can occur on either strand. It could occur here, and then you would read from 5' to 3' could occur here. It could also occur here. And so that's a nice feature of DNA that one has to keep in mind. So yeah, that's the essentials of molecular biology. There are actually three common biomolecules we talk about: DNA, which we just talked about; RNA, which is a single-stranded molecule, which has a different sugar. It's like DNA, but instead of deoxyribose, it has ribose, and instead of thymine, it has uracil, so it has a different nucleotide. But in a way, it's like a one-on-one match with DNA. So it is used partly as a messenger molecule, that a DNA molecule can be transcribed into an RNA molecule that's got the same sequence. And that RNA molecule typically might code for a protein, or it could, RNA molecules can have their own function. So DNA carries genetic information. It basically contains these coding regions. It also contains non-coding regions. Most genes code for proteins, but some also code for RNA molecules that have their own role. And so I won't talk about proteins very much, but essentially there is a three-letter genetic code that translates RNA to a sequence of amino acids. And there's a machine called the ribosome that does it in the cell. And the protein, once it is made, or in fact, as it is being made, folds up into a complicated 3D structure. So some proteins are enzymes that are catalysts and health reactions happen and so on. Some proteins are structural. So there are all kinds of proteins. Proteins are the building blocks of life, basically. But that's kind of all I'll say about that. And I'll skip this part. As I said, I have a lot of slides, but I want to focus on the algorithms here. Okay, so I talked about DNA and sequence. Now, how do you get the sequence of DNA? The reason I'm talking about this will become useful, but essentially, to get the sequence of a DNA molecule, you have to read it one letter at a time. How would you do that? So the first generation sequencing was invented by a guy called Frederick Sanger and also Walter Gilbert, and they got the Nobel Prize in, I think, 1980 for this. It uses something called gel electrophoresis. It's a very slow process, but it's very accurate. And you cannot read the entire genome of any organism at a time. So just for sense of scale, bacteria like E. coli might have 3 to 6 million base pairs in one single chromosome. They just have one chromosome, which is circular chromosome. Um, Humans have 23 chromosomes, 23 pairs of chromosomes, you should say. And the total content of the longest of these is about 250 or 300 million base pairs. And totally, they amount to about 3 billion base pairs. And so that's the kind of data we're talking about. And there are organisms that have much bigger genomes, including some plants and even some things, similar amoeba. So it's not clear why they need to have such big genomes. And within the human genome, we only know about 2% of it goes for proteins. And about half of it is repetitive stuff, and we have no idea what it's there for. There are theories, but it's really complicated. And some of it is regulatory, like I said, as in it helps in turning genes on and off. Some of it helps in packaging the genome so that it behaves sensibly. But still, we don't know most of the genome, what most of the genome does. So the first generation was just solid sequencing, and you read 10,000 base pairs at a time. And so you to sequence the human genome. So in the 1990s, this was the technique that they had. And both the NIH in the US and independently Celera Genomics, which is a private company, wanted to sequence the human genome, and they combined forces, they actually did it. And some of the success came from new algorithms that they. So basically, you got lots of these string leads, they call it, which are a few tens of thousands of base pairs long, but you have to assemble them into one big genome. It's like a jigsaw puzzle, but it's not quite like a jigsaw puzzle. We'll come to that. Second generation continues till today, so mostly driven by this company Illumina, and essentially they are able to sequence lots of the genome in parallel and also tell you some kind of quality score of how good their sequencing is. And on the flip side, their reads are much shorter. It's about 30 to 200 base pairs. That's still kind of stable young today for most people. The cost has come down sharply from 2005 to now. There is a third generation which is beginning to get active, which is a much more compact device called the nanopore. And this gives you very long reads. It can give you, again, thousands or even, I think, a million base pairs. But the trouble is it is kind of got a high error rate in the range of 5 to 10%. I think it's come down a little, but it's still more than 1% error rate. So it is useful for some purposes in combination with the other method. So I guess I won't talk more about that either. So these are the tasks that you have with next generation sequencing. The first one is assembly of genomes de novo. That means you don't know what the genome of a new species is. You found a new frog or something and you want to get its genome. You put its DNA in an Illumina machine and it gives you these reads, which are 30 to 200 base pairs, and you have to stitch all of them together to get a genome. That's a very complicated task, but some of the basics of this are quite simple and interesting. Then the other thing that people do with next generation sequencing is probe various aspects of what's actually going on in our cells. So one example is which genes are being expressed and at what level. And so genes, as I said, are basically transcribed into RNA molecules. And you can sequence those RNA molecules also. Basically, they reverse transcribe them back to DNA and sequence them. So this technique, RNA-seq, does that. And from this, from sequencing those RNA molecules, you can tell, you can map back to the genome and see what parts of the genome they came from, so what genes are being expressed. This technique tells you what proteins might be binding to DNA. I won't tell you how it works exactly. It's called chromatin immunization followed by sequencing. But essentially, you are able to pull down parts of the DNA that bind a protein that's interesting to you. And then you can break it off and you just have the pieces of DNA that are bound by a protein. You can sequence them and map them to the genome. And that tells you where in the genome those proteins are binding. both of these tasks and many other tasks in this require you to map short strings, short in the sense of about 100, 200 letters long, to a longer string which can be 3 billion letters long. So this is a very standard problem in computer science called finding patterns in text or substring finding. Have any of you written a program to do that given text T? Okay, let me look for us. Yeah, exactly this. These are two possible problems. Let's focus on this one. Given two strings S and T, can you find all the places where S occurs in T? Substrings, yes. So, no, I mean, this, you're given that S could be a substring, you want to find where it is a substring. S is a substring or S is a possible substring. If it doesn't occur, you say it doesn't occur. If it occurs, then you say it occurs here. The other task is, so this is the task that the ChIP-seq, RNA-seq and all that would do. You have reads and you have a human genome, a draft human genome that's around. You take the read and map it to the human genome. Where has it come from? So that's a version of this problem, except T is very, very big. And the other problem would be finding overlaps between strings. So supposing this is one string and this is another, and yeah, so how would you find an overlap between this? So this piece is the same as this. So in a way, they are kind of similar problems. You're finding patterns that occur in both strings. So this problem would be useful in assembly. That is, you've got a lot of reads and you want to stitch them together to make. So how would you do? How would you address either of these problems? The other third problem could be common substrings. That means if so, I won't go into this, but there's an example about it. Supposing you have S is this and T is this. S does not occur in T. S is not, there is no common prefix or suffix of them, but is there a sequence that occurs in both? So for example, by looking at it, you might say that this, not that, this piece occurs in both, right? But how would you tell a program to do it? Or how would you do it even manually? So since the 60s or 70s, people have been looking at algorithms to do this one. So yeah, so I'm going to borrow heavily from slides from this guy when I need to make his teaching material freely available. So wherever I'm using the slides, I've got this line at the bottom. So let's go with some. So before I continue, any questions, comments up until this point? So just some definitions. A string in computer science is a finite ordered list of characters, meaning, so you have an alphabet, which is conventionally written as sigma, and well, ignore the parts you don't understand. Order one means that you don't care how big the alphabet is. This one has 4 letters, this one has 20 letters. The English alphabet has 26 letters. So a string is just a sequence of characters drawn from some alphabet. And the length of string is written as S is written as this, it's the number of characters in the string. And epsilon could be written as the empty string, so its length is 0. So if you're writing in Python or something, you will write the empty string as this, just 2 quotes next to each other with nothing there. And yeah, so for 2 strings S and T, we'll define the concatenation as everything in S followed by everything in T. So concatenation just means joining together. So you have like you have 2 strings A, T, C, A, and the second string is C, A, G, C. You just write both of these as one string, and that's the concatenation. And S is a substring of T, and you will write them as just ST, the concatenation. Now, if it's a substring of T, then there are possibly empty strings U and V such that T is U concatenated, the rest concatenated with V. All that means is that T occurs somewhere, S occurs somewhere inside T. Maybe S is equal to T. If we are both null, then S is equal to T. Maybe S is a prefix of T, that means that V is null, that U is null, or S is a suffix of T, that means that V is null. So these are basic definitions. And subsequences. Okay. But how would you find an exact match of a word? So this is called an, so let's say P is the word, it's literally the word in this case. And you want to ask where in this text does this pattern occur? So that's kind of the most basic problem in pattern matching. So how would you do it manually? But essentially, you can place P anywhere you like and check letter by letter, does this match, does this not match? So supposing P was placed right here at the beginning, you will compare W with T. No, it's not the same. So it doesn't occur here. But supposing you placed it here, you would have the O, R, D. But you compare W with W, it's the same. You compare O with O, it's the same. And you compare R with U, it's not the same. No, it doesn't come here. So if you do this position by position, eventually you will find that this matches and you'll get your answer. So this is kind of the simplest way to do it. But is it the fastest? So here is something that computer scientists worry a lot about. How much time would your algorithm take on average in the worst case, maybe in the best case? So let's say the length of the pattern is n, and let the length of the string be t. Then this algorithm, as we just described it, we are comparing one letter at a time. At each position, we are comparing at least one letter, right? So, and we are scanning the entire text. So the number of operations you need is at least, yeah, it is O and M time, yes. And that's the worst case. In the best case, it's better. And the reason for that is that the length of your text is n, and you're scanning the entire text, so you have that n. But at each position, you're checking a few letters. Now, in the best case, you're only checking the first letter, and only when you reach the end, you'll actually, for example, supposing the letter W never occurred anywhere in the string. In fact, it occurs only here, except for here. So, in the best case, you will not be checking the length of the pattern very much, only the first part of the pattern. But in the worst case, you could actually be checking the whole pattern every time. And here's an example of that. Supposing P is equal to A, 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 T. And the text is A, dot, 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 full of A's. Now, this is an example of the worst case. So this is patterns of length 6, textures of length, let's say 1,000 or a million or whatever. But every time from this algorithm, you are checking all 6 letters of P and finding only at the 6th letter that it does not match. And then you're moving one step, and then you're moving one step. And so this will basically give you the worst case running time. So O, big O, NM, for those who don't aren't familiar, it means that the number of operations that you need to do this task is less than some number times n times m. And that number could be kind of big, but and there is no so you could have order of n times m. So let's say n is equal to 10, m is equal to, let's say, 10,000. Then this means you'll require something like 100,000 operations. It could be 500,000, it could be a million, but some number times 100,000. But there is a constant number which can serve as an upper bound. But if you write n plus m, this grows less fast than n times m. And essentially you cannot make a claim for this algorithm that it is order n plus m, because there is no number you can say such that your running time will always be less than that number times n plus m. As you increase n and m, it will always overshoot any number that you. So that's how computer scientists think about this, and it's almost all the papers you read in computer science will be how to reduce the bounds in running time of some algorithm. So this is the simple algorithm that we just align. You try all possible alignments each time you check, and that's 9. It is ONM. It kind of depends on your problem, I guess, but mostly you will check. It may not be ONM, it may be OM, but it depends on the problem. So it depends on how big your alphabet is and how big how random your sequence is. So in real applications like the anyway, thank you for questions, and the rest of you also please ask. DNA is actually not very random. You have lots of repetitive stretches and lots of repeated letters, and you have only 4 letters to work with. So this kind of an algorithm will come close to its worst case from DNA. Right. So you can actually improve this a little bit. Yeah, so basically the greatest number possible is this, and the least possible is this, because there isn't any occurrence of W except in the actual match, then this is it. So you can improve it a little bit, because so now you were looking at this W and this O also matched, and then you are trying to match R and you see a U. Now you know that it is not matching here, but you also know that the letter U doesn't occur in the pattern that you're trying to match. Therefore, you don't have to bother with matching this position or this position, because you know U is not there. You can skip right ahead to this position. So that improves things a little bit. It turns out you can improve it quite a lot more. And there are 2 algorithms that were written in the 70s, Ruth Morris Craft and Boyer-Moore. And Boyer-Moore is kind of the fastest algorithm of its kind when you are just given a pattern and a text. So I think I'll skip past this. You can look it up on Wikipedia or something, or on the guide site if you like. But essentially, you have these 3 rules that allow you to skip to great length. So one of the first clever thing it does is that it starts the matches from the end of the pattern and not the beginning. So you align the pattern here, but it's not looking at these characters first. It's looking at these first. And what that does, I won't go into the details, is that when you see a mismatch, it allows you to skip ahead by sometimes quite a bit. And so if you have seen a mismatch here, you don't have to slide it once, but you can actually slide it all the way here. And then each time you're matching going backwards like this. So only when you actually find a match, you end up comparing all the characters, but most of the time when you find a mismatch, you just jump quite far ahead. And the net result of that is, so there's a book by Dan Gusfield called Algorithms and Strings, Sequences and something, and it's worth reading if you're interested in this subject. Gusfield actually visited CMI a few years ago. An interesting guy to talk to. Yeah, so I'll skip the details of this algorithm for now. But the worst case of Boyer-Moore is order M, where M is length of the string. But the best case actually is floor of M divided by M. That means you have a text of, let's say, 1,000,000 and a pattern of 100. Then you actually need only 10,000 comparisons. So that's pretty remarkable. Can we do even better? So in both these cases, so in Boyer-Moore, you actually have some pre-processing for the pattern, which doesn't take long because the pattern is small, but you make a lookup table and so on. And you do it once, and then you can use that same pattern to match multiple texts. So for example, you're looking for a string in human and also in mouse and also in fruit fly. You could do that. Not really. Yes, but that is a constant. You're doing it once and it's only n. So, so you could say order m plus n because n is the pre-processing time. But yeah, it's just n. It doesn't depend on the text. It only depends on the pattern. No, it's just n. Yeah. So it's just the kind of matrix that you put up. So, so I don't think it's there in this, but you can look it up. Yeah, size half of it is also there. We often ignore that, but it is. Okay. Right. So in terms of actual running time, for this guy's implementation in Python, which is a very straightforward implementation, the live matching takes okay, so this is some simple texting, and here the difference is not so stark. Here the difference is it's really not great, but if you do boil more efficiently, so the grep command in Unix, Linux, and then in particular the version of grep that's in Linux is extremely fast. That's partly because so it uses boil it does a whole bunch of other tricks that help it to search very efficiently in large files. So if you want to find where a particular string occurs in your entire mailbox dating back 20 years, you can do it in like very quickly. Okay, so how do you get better than this? So one way is you are trying to search for a pattern in a text, and the pattern is small and the text is big. But supposing the text is kind of given to you, like it's the human genome, and it's there. So can you pre-process the text in some way? And maybe it takes you a bit of time, it doesn't matter. But having pre-processed it, you can search as many patterns as you like in it. And supposing the pre-processing time has to be at least order of the length of the text, but you do that once. But then the patterns you've got are all these reads from some sequencing experiment, but millions of them. But for matching each pattern, suppose you could do it in order m, the length of the pattern. Turns out you can. And I'll sort of quickly overview particular data structure called a suffix tree that is widely used for this. Suffix trees were invented in the 1970s. And once you have a suffix tree, it was very clear that you can search in linear time. So I'll explain how that is done. But it was only in the 1990s that guy called Yokonan showed how to construct a suffix tree in linear time. Not just in linear time, but online, meaning as you read a letter, you can add it to your suffix tree and you end up with the whole thing. And that was kind of beautiful algorithm, but it's too complicated to explain here. But that kind of revolutionized genomics that it suddenly enabled you to do this kind of magic. I'm not very sure what Google does because the drawback with suffix trees is that it does take up a lot of space. It's linear space, but the prefactor is large. So even in bioinformatics, suffix trees are used for smaller organisms, but for something like human genome, it becomes these days it can probably be done, but I don't think it's doing it. The more complicated data structures that they use. If I have time, I'll mention those, but I might not get to that. So let me just talk about suffix trees. Before suffix trees, let's talk about trie. It's pronounced trie. It's a tree representing a whole collection of strings. Supposing you want to store a bunch of strings, a bunch of long words, if you like, in some way, so trie stands for retrieval. You can retrieve things that are stored there, and so here is an example that you want to. So it can be used, for example, for as a dictionary, as an implementation of a dictionary, where you have a key and you want to retrieve a value from the key. So let's say these are the three keys. So a trie is the smallest tree such that each edge is labeled with a character, and each node has at most one outgoing edge with that character. and each key is spelled out along some path. So, I mean, label C for any C. I mean, a node can have two outgoing edges, but only one with the letter S. So how does that work? You can construct a trie like, so if you just have one word, then that's it, that's your trie. So it will be I, N, S, T, and so on. And that's basically this path here. When you're storing your second word in the same trie, as long as your letter matches, you keep it. When you have a new letter, you have I, you have N, but now your word is internal. So you need a new branch here and continue like that. So that's basically the idea. And instead of storing 3 words of length, in this case 7, 8, and 9, you are basically storing each letter exactly once if it's unique. So it's some amount of space saving is there. And you can quickly retrieve the words that you've stored. So if I now give you a new word, so this is a case where you only store 3 words, but you could store 300 words. And that's the list of words that you're looking at. Now I give you a word and ask, is it in your list? You can quickly check this and see if it's not there or not. So if you can traverse the trie and reach a node, you can find, you'll say, yes, it's there and its value is this. If I gave you the word international, I will go INTERNA, and then I only have an auto L, but I want a T. So at this point, I will say no, international is not specified. So that's the basic idea. And you could have try that where the words that are stored are suffixes of your text. So you have a text like banana. What are suffixes of this text? They're basically all strings such that this text is equal to you concatenated with S for any U. So you could be a null string, so the entire text itself is a suffix of itself, anana is a suffix, nana is a suffix, and so on. Supposing you chose all these suffixes of a text to store in a trie. Why would you want to do that? Because I'm doing pattern matching. And now, if a pattern P occurs in text, then the pattern P has to be a prefix of a suffix. Is that clear? So, for example, let the pattern be NaN. So this pattern has to be a prefix of a suffix of the text. And in fact, it is because it is a prefix of NaNA. It is a prefix of this word, while this word is a suffix of the full text. So if I store all these suffixes of my text into a trie, then I can check for any pattern by seeing whether the pattern is fully spelled out along a path. I don't have to reach the leaf now. I don't care about these numbers now. I just want to know whether this pattern is there. So if so, let's move on. Yeah. Yeah. So this is the example of a suffix trie. So this is your text. Now it's got only 4 letters, ABCD. And these are all possible suffixes of this text, ranging from the full text to just 1 letter, the last letter. And if I were to search for a pattern, I just need to check whether it is a prefix of one of these words. So what do? I build a trie. The first thing I do is actually find a special terminal character. And the reason is that I want some kind of unique ending. So it guarantees that no suffix is a prefix of some other suffix, because this dollar is a unique character that only occurs at the end of string. It doesn't occur in your alphabet otherwise. So that's just a kind of trick you use. And then you can make a try like that. Okay, so making this a try for this might be too complicated, but let's make a suffix try for this text. So ABA, you add a dollar, so ABA dollar, and this is the try. So if you check it out, all suffixes of this are spelled along some path. So um, A, B, A dollar, the full string. The empty suffix is just this guy. Um, A dollar is this guy. That's the last suffix. So everything is there somewhere. And you build it up basically by starting from the longest suffix and then continuing and adding more and more, or you can start with the shortest suffix and add them. So you can now search in this for any pattern in linear time, right? So you can now think of nodes as having labels where the label spells out the characters from the past to the root. So you can think of this as, so it's not explicitly stored here, that would be the point, but this is what you would get if you spread this out. So if you can spell out any string s along some path here. So how do you spell it out? Your string is ABA. No, your string is BAB in this case, I think. No, BAA. So this is your string BAA there. B is there, A is there, A is there. So it's good. So it is a substring. Is your string BAB there? B is there, A is there, but there is no path B going out of it, so it is not there. So it's not a substring. So that's basically how you use it. And the full string itself, ABA is a substring. BAABB is not a substring because you follow, and so on. So how do you can also check whether the string is a suffix. The same thing, except you have to reach the endpoint. If you haven't reached the endpoint, then it's not a suffix. Okay, so I hope that's kind of clear, but just looking at this, it looks like it makes a lot of space, right? So how much space would you need? I think that was an exercise for you. But you can crunch up a suffix try quite a bit by, yeah, so this is the size of the suffix try as a function of the length of j. So it's actually goes as somewhere between m and m squared. It's not quite quadratic, but it's pretty bad. How can you crunch it up? The question. So basically, the two ideas that go into making it more compact is when you have a chain of nodes like this where there are no branches, everyone has only one descendant. What if you just replaced all these nodes with a single link where now instead of a single letter here, you had an A, you had a B, you had an A, you had a dollar. Instead of that, you just have one link with this entire thing A, B, and dollar. You don't lose any information doing that. So that's the first thing you do. And so having crunched it up, all the internal links, some of them have multiple letters now. But now instead of that huge tree you had here, you have this much more compact tree. So this is called a suffix tree. You can still save space. So the problem with the try was the amount of space it requires. And what is the space this requires? It is the total length of the edge label is quadratic because you are now storing the entire label here. You have to store it somehow. And some of these labels will be of the order of the length of the string. So, and you have that same number of tables. So you have something of order m squared storage required. So the next idea for suffix trees is you don't have to store the labels. You can store the two numbers, the starting index and the ending index in the string where that label occurs. So you store the text itself. You keep a copy of the text that is of order m, and these labels become offset length pairs. That means ABA dollar will be, if your offset starts from 0, then this will be 3, and its length is 4. So you store ABA dollar at 3, 4. You store, let's say, most of them are something comma 1, this is 1 comma 2, that is the DA. This thing, it used to be DA, now 1 comma 2. So that's the basic idea. Now you can store the suffix tree in order m space. So you have 3 billion letters of the human genome. You can store them in order 3 billion bytes. But order means there is still some number in front. And that's the problem with suffix tree, that the number, because it's stored as a tree, ends up becoming pretty big. Plus, you need to store all these numbers. So you need 2 integers per link and so on. So it works very well if your text is not too big. Yeah, the last part, so I'll skip this part, I think, but that's the basic idea of a suffix tree. It is actually very widely used in some contexts, but you can do even better. And the building part is where you can make this contribution. And there are many applications for this. So you can find longest common substring, longest common the generalized suffix tree is where you can store 2 strings. And the way you do that is you have 2 terminal characters. Okay. I have time till 10:30, 10:45? 10:45. Okay. Yeah, so I'll skip ahead to a couple of other examples of algorithms. So a suffix array is one example where you store all the suffixes of your list of your sequence in lexicographical order, that means in dictionary order, with the convention that dollar is the smallest. So your first letter is always dollar, and dollar precedes any of your other letters. So with that convention, you store all suffixes in dictionary order, and then you store the index of each suffix. That means where did the suffix occur in the original string, 0, 1, 2, 3, 4, 5, 6. So 6 is the last one, and 0 is the full string, 1 is this guy, BA, dollar. So, and then you throw away this part. This is of order m squared, but you don't need it. You keep only this, which is of order m. It's just a list of m integers, right? It's just where those suffixes occur. Then if you're given a pattern, how do you find whether it occurs there? So again, your pattern has to be a prefix of some suffix. So you take the first letter. So yeah, it's a little bit clear. So yeah, it must be a prefix of at least one of these suffixes. And the suffixes that share a prefix are consecutive in the suffix array. So you can actually use binary search. So I don't know whether you're familiar with binary search. Essentially, if you have a sorted list and you want to find where something occurs in a sorted list, one x2 dot x n. If this is a random set of lists and I'm asking you with this item in there in the list, then you have to look at every one of them one by one. So it's of order n, right? But if I'm telling you the list is sorted from lowest to highest, then to check whether the item is there, I first check the middle guy, x n by 2. Is it equal? Then I just return it. Is it greater? Then I search this half of the list. Is it less? Then I search this half of the list. So each time I divide the space I'm searching by 2. And as a result, the number of comparisons I need to make is not n, but log n, log to the base 2 of n. The base doesn't matter because it's order, so the base will come out in 3 factors. So you can use binary search, and that will require order log m operations. And so that's slower than the suffix tree, because there it is just n, but here it is n log m. But log m is not so bad, and the space that it requires is still pretty small. And there is an even more interesting way to do this that I won't go into now, but you can look it up. It's called So some of you may be familiar with something called the Burroughs-Wheeler transform. It was originally invented for compression. And it's basically the transform is take all the permutations of your original string, cyclic permutations. So meaning you can start anywhere you like and go back till the previous letter. So these are all the permutations, sort them lexicographically as before. And then the last column here is your Burroughs-Wheeler transform. Now note the link with the suffix array. The suffix array is just exactly these guys, except you stop at the dollar. Those are all the suffixes of your tree. But here you're keeping the entire rotation, and then this is your transformed string. And it turns out that this is easily invertible. And the last column tends to group similar letters together. So you can do run length encoding to compress the last column. And because it's, so there's a program called bzip2 that is quite popular as an alternative to gzip or zip and so on, that uses Burroughs-Wheeler transform. So it's useful for compression because the last column tends to have letters, the same letter repeat itself. And so that helps you to compress. It's reversible. I won't get into that, but and how this use as index that also I won't get into, but let's just take it that it can be done. So with that, I'll move to, I think, the last topic that I want to cover and then break for questions. And that topic is genome assembly. So far we've been talking about mapping. That is taking a short string and mapping it to a larger string. Now you have something like you have lots and lots of strings. And so I'm going to switch to a different set of slides here. Sorry. Yeah. So you can think of the genome assembly problem as exploding newspapers. This is from a book by Compo and Webster. So supposing you have a stack of newspapers and you sort of apply some dynamite to them and they explode. Don't try this at home. But what you have is a whole bunch of fragments like this, and the task is to reconstruct just one copy of that newspaper. You don't want all of them because they're all identical. So the analog here is that you have a genome, you have multiple copies of genome, you have a whole bunch of cells, you have thousands of cells maybe, and you're running them through a sequencer, and the sequencer is sort of chewing up your genome. It is giving you short reads, and it's not giving you continuous reads from any one genome, but it is giving you enough reads that somehow you should be able to piece them together to make up the whole genome. That's your task. So it's an overlapping puzzle. So you have to search for pieces that have an overlap, like this piece overlaps with this piece, and so you can combine them together. And if you keep on doing that, eventually maybe you can piece together the entire paper. So this is the analog for sequencing, that you have multiple copies of the same genome. In this case, 4. In real life, it will be many more. And the sequencer is sequencing them, breaking to random positions. It is not even necessarily, in fact, it is not just breaking it up, but it's also losing pieces. So a lot of the, a given read will, a given sequence will have, so the way it actually works is that you have a breaking down sequence step. The genome is broken down into pieces of roughly 500 base pair maybe. But then your sequence actually, sequencer only reads the first 100 base pairs or so. So the rest of it is kind of lost. And there is a version called paired end sequencing where you read the other strand. Since DNA is double stranded, you can read one strand from here and one strand from here, but I won't get into that now. We just have one strand to deal with. But we still have this jigsaw kind of puzzle that you have lots of these short reads, and you are hoping that they have enough, they call it coverage, that any part of the genome has been sequenced at least X times, like at least 10 times or 30 times. And so this is the data you're given. You have to sort of piece them all together into a genome. And that's the overlap task. That is, if you have any one read, you want to find out which other read might overlap with it and take these two reads. So if you took this one, you might search all the others and find that this one has an overlap of 5 letters, 6 letters, and so you stitch them together. And so that's kind of the goal here. So I'll maybe pause to say that, okay, so let's go through this a little bit more. So naively, basically what you need to do is what you have is a bunch of reads. So take this example where you have, this is your original string, and these guys have given an even more toy problem of it, that they're telling you that if they got every theme from this string, so you've got A, UAT, ATG, and everything is shown up as many times as it actually occurs in the string. So ATG occurs 3 times. Given those reads, can you reconstruct the string? So what you have is just a bunch of reads, and you want to make links between those reads based on their overlap. So this is called the overlap layout consensus method. And that's just to maybe illustrate it, you have a read which has TAA, you have a read that has, in real life, the reads are much longer, like 30, 40, whatever. But let's say you have just 3. You have to somehow identify that the suffix of this is the prefix of this and join them together. And so what you have is a graph of reads and links, edges between those, directed edges between those nodes that occur when the last k-1 letters of this is equal to the first k-1 letters of this, then you put an arrow. And then at the end of the day, you have to walk through all the nodes. You want kind of the shortest string that can represent all these, the shortest string such that all of these are substrings of this. That's the task. So you want a path that walks through all of these in the, in the ideal case exactly once. That's called the Hamiltonian path. You have a graph, you have nodes and you have edges, and you want to walk through the graph, visiting each node exactly once. It turns out that's a very hard problem to solve. It's one of the problems that's called NP-complete, if you're familiar with that. The idea used by modern assemblers is to reframe the problem a little bit. For that, I'll so slight switch of topics. Have you? So this is a sort of pseudo Sanskrit word that I'm going to write down here in English. That was, I don't know whether anyone, don't try to read it as Sanskrit, but the key thing here is that small a is what they call Laghu A, and the big A is the Guru A. So there are 2 kinds of syllables in Sanskrit, short and long. And this comes from Sanskrit prosody. There was a guy called Pingala 2000 years back who made some roots for it. So how many 3-syllable combinations of short and long can you make? And the answer is of course 8. So you could write it as A. Let's write it like this. Now there are these 8, and he gave some names like this was Napada and this was forget. I mean, each of these have some name, but this was basically, now if you combine these just the vowel sounds of the string, this is a string of 10 letters. Now, if you ask what is the shortest string where each of these occurs as a substring, this is an answer to that. It's not the answer, it is um, an answer. And it is a special case of a problem that Dutch mathematician called de Bruijn raised in the 20s or 30s, I think. That is, given an alphabet sigma, here our alphabet consists of just two letters, small a and big A. And given an integer k, what is shortest string S that contains as a substring? And his problem was a little bit more specific, that what is the shortest circular string? So what's a circular string? And you join up the tails in itself. So you notice that the first two letters here are actually the same as the last two. You can actually join it up like that. So let's write it like that. And you go in this direction. So each, so this is just 8 letters in a circle, but each of these 8 words occurs somewhere in these 8 letters. And so the more general version of this was given k words of length k and an alphabet of size sigma. So for DNA, the alphabet is ACGT, it's of size 4. Can you find a short string that has every k word as a substring? So that was his problem. It's been adapted to assembly in a way I'll just get to. But how did he solve this problem? He solved this using something called a de Bruijn graph, where every node has the k minus 1 merge. So let's say you have, so you have only 2 letters in this case. So there are only 4 possible k minus 1 merge, right? Where k minus 1 is equal to 2. So it's AA, small a, big A, small a, and big A, big A. These are the only 4. And now you put the themers that you're cared about on the edges of this. So you make edges between these such that if there is an overlap between these, the last letter of this and the first letter of this, then you can put an edge. So there's one like this, but there's also a self edge like that. And those are the only 2 possibilities from here. Here the last letter is big A, so it can go here, or it can go here. And that's it. This is also big A, so it can go back. It can go, so the last letter is small a, so it can go here, or it can go here. And the this guy has a big A, so it can go back to itself, or it can go here. So those are the possibilities. So this is your graph. It's called a de Bruijn graph. And now your task is, and let's explicitly write the, well, so this edge corresponds to small a, big A, because that's what you get when you go like that. And this edge corresponds to small a, big A, small a. Sorry, no, not this edge. This edge corresponds to big A, small a, big A. And this is small a, big A, small a, and so on. Now you want to go around this graph, returning to the same starting point if you want a cyclic string, visiting each edge exactly once, not each node, but each edge. So that's a problem that was first addressed by Euler. There's a city in Russia called Königsberg. It was in Germany at that time. Now it's called Kaliningrad, and it's not over sure. But that town had, I think, 4 landmasses and 7 bridges. And the question was, is there any way to walk around the town, crossing every bridge exactly once, coming back to the same starting point? And Euler showed that you can't. There are slides for that also. Yeah.
The AK, would you take use of K minus 1 and match the last K minus 2 letters?
So if it is not the case 4, for example, you will match lot 3 in this. So the case, case 4 k minus 1 is 3. So you match the last 2 letters with the first 2 letters, and you get a 4 letter. Sorry? Yeah. I believe it's pronounced Dip Brown, but so when I switch back to the slides now, was that bridges of Königsberg problem was here. Sorry, yeah, so this is the map of Kaliningrad, and these are the 7 bridges, and you can map them as the graph. Now, the key insight is that if you want to get back where you came, then the node and every node must have an even coordination number. That means as many ways to go as to come in, because every time you enter a node, you have to leave it by a different bridge. You cannot go back to the same bridge. So you must have 2 or 4 or whatever. Now, there's a version-less problem where you don't have to come back where you started, but you still have to visit each bridge once. That means that you can have one bridge that has you can have 2 bridges that have an odd coordination number. You start at one and then the other. Everyone else must have an even coordination number. Now, in this case, every node actually has an odd coordination number. It is 3, 3, 3, 5. So there is no way that you can do either an open or a closed loop on this, because you cannot have more than 2 nodes with odd coordination number to do this. So that's basically the reason why Eulerian works. So you can verify whether a problem is solvable or not quickly just by looking at the coordination numbers. You can also solve it in linear time. A Hamiltonian walk where you have to visit each node exactly once takes a long time, but an Eulerian walk can be done in linear time in a number. And the way that is done is you can take a walk. If you get back where you started, you take another walk and you kind of fuse all of them together. So there's a bookkeeping way you can do that, where you can combine a bunch of different walks into one long walk. So that's basically the way people assemble genome now, except that the ground graph that I mentioned was for the shortest string that contains all substring, all k-mers. Instead of k-mers, you can have substrings of the reads that you got from your genome. And instead of asking so it will not be all possible k-mers, it will only be the ones that actually occur in your genome. And then you construct a Hebron graph, and you use that to you find a walk around it, and that basically assembles the genome. That walk is the assembled genome. So now there are many complexities here. Just this by itself never works, because for one thing, the genome is highly repetitive. For another, sometimes your reads have mistakes in it. All kinds of things happen. So I'll kind of stop here, but these are sort of some nice toy problems in computer science that actually have, biology applications in the real world that are used today in serious problems. So there are 2 messages from this. That one, all these nice algorithms are actually useful, but 2 is that to use them on real genomes, it's not always as simple as that. You need to do, you need to take account of errors, you need to take account of non-ideal situations, and so on. So I think this is a nice slide to leave it on. So I'll stop here. And, yeah, thank you very much. Yeah. Okay.
So what do you do in case of errors? Because I imagine that you can take it very on purpose.
Let me see if I find that last question there. Sorry. Even this, yeah, so this is an example. So before errors, the other problem actually is that can have multiple paths, multiple solutions to the oil problem. And so if you look at actual genome assembly results, very often they cannot tell. They can tell that these leads came together, these leads came together, and so on, but how they arrange. So they report an initial genome as a bunch of contigs, and then they use other ways to stitch those contigs together, and it can take years to get the right answer. Now, in terms of errors, what happens is, suppose you have a single read that had an error. This is an example here. So these are the reads that you actually got. And one of those reads had this C, which was wrong. Now, the way the Deborn graph assembly works is that you break the reads further into smaller schemas. So you don't do the Deborn graph with the reads themselves, but with some smaller schema, like 10 or 12 or something, instead of 30. Now, because one of the letters there is wrong, a bunch of your schemas will be wrong. And that basically shows up like this. This is the correct path. And this is an alternative path that came only because of your problem. But the other thing is that in a real problem, you have a heavy amount of over sequencing, 30x or 60x and so on. So once you break it up, so your original reads might not be identical. They are like 30 or 100 letters long, but you're breaking them up into schemas of length 10 and then making this graph. Now, those schemas of length 10 will be very repetitive because your read, one of them might have been like this, the other might have been like this, but the length 10 schema will occur in both of them. So when you weight these arrows by the number of reads you get, this will be a much heavier path. There'll be many arrows here. Just one read is wrong, that will be like an alternative path, but it will be a much lighter arrow. So basically, this is here the heavy arrows are the okay reads, but because of the errors, you have all these problems which you can ignore because you know that only a few reads happen. Okay. Yeah.
Could you elaborate a little on how this is an NP hardware? Because I imagine if you have
okay. It just is because it is NP complete because they prove that it is. NP complete means that so if you can solve it, then you can solve all NP complete problems. But why it is maybe let's say it's just a combinatorial thing that so how do you
for the general having part of the NP
Yeah.
complete that I think here in the graph I feel like if you go like you, how do you make your graph by just saying it's the first K, last K of this one is the same as the first
you're saying that this graph have some properties that make it simpler. That is a good question, and there are people who argue. There is a paper by Gene Myers and others who say that for the specific kind of overlap graph that you get, the Hamiltonian path actually can be done in linear time. But that is a claim they make. I don't think there is anybody who has implemented it in the real world successfully. So maybe it can be done. I mean, Gene Myers thinks it can be done, but he hasn't done it. And yeah, that's a good question, but yeah.
You mentioned about online analysis.
Yeah. Yeah, there is a normal algorithm by opponent for a suffix tree. So let me maybe scroll back to the so how do you build a suffix tree? You can naively make a suffix tree and then crunch it up, but that will initially require order in squared space. And but it can be done online because as you actually I'm not very sure how this cannot be done online. I just need to sequence to do it. So I have at some point tried a method, but I don't remember how it works. It's kind of a clever method, but I don't think this is your algorithm. Essentially, online means you're reading one letter at a time. and you're putting it into the tree as and when you need it. But yeah, it's that means you're kind of also growing the suffixes in the tree as you're reading them. And it's so yeah, how it does it, I can't quite remember. You can look up Custill's book, is probably the best explanation.
Are there any tools or advanced and how to evaluate?
This is already linear algorithm, so I don't think you can get better than that. I would guess so. Are you based in Yemen? There is a guy called Venkatesh Raman in my institute who is an expert on chemistry. He might be the right person to talk to. There's something called compact substrate or succinct representation of substrate, all about improving the constants for the storage part of it, not so much the construction, I think. Thank you very much. Yeah. Yes. Next in the plan.